One of our research projects was the production of a therapeutic biosimilar. Bevacizumab, which is marketed as Avastin, is a humanized monoclonal IgG1 antibody against human vascular endothelial growth factor A, or VEGF-A. Bevacizumab works by inhibiting angiogenesis and has been used clinically to treat several different forms of cancer for many years. It is normally made in traditional mammalian cell lines, but here we were able to produce a Bevacizumab biosimilar inside the leaves of plants. Our antibody was produced using two separate plasmids containing the heavy and light chain of the antibody sequence. These plasmids were co-infiltrated together into the plant leaves, and the proteins produced in parallel within the plant cell. The antibody fragments naturally come together in the plant cell, forming the full antibody complex. We were able to optimize our upstream expression processes by identifying the ideal day for harvest, maximizing the accumulation of complete antibody within the leaves of the plants, and through the use of a plant signal peptide, which improved yields by localizing the antibody to a specific organelle within the plant cell. Downstream processing of the infiltrated leaf tissue was carried out in large-scale batches of up to 1 kilogram at a time. We took advantage of the relative size of the antibody and used tangential flow filtration to separate the antibody from the bulk of plant host cell proteins and pigments. In several steps, this involved microfiltration and ultrafiltration. Finally, purification of the antibody was carried out using several classic chromatography methods, producing a pure final sample at yields on average of around 200 milligrams per kilogram of leaf tissue. We tested the activity of our antibody biosimilar against its target, VEGF-A. Binding of the plant-made antibody to VEGF-A was analysed by surface plasma resonance. We immobilised a human VEGF-A onto an SBR chip and then increased the concentration of antibody that was flowed over the chip. A binding response was measured over time and at different concentrations of the VEGF-A. However, due to strong binding and therefore slow dissociation rate of the antibody from the immobilised VEGF-A, the kinetic parameters could actually be calculated from a single cycle kinetic experiment. And the calculated values agree closely with those quoted in the literature for mammalian-produced Bevacizumab. We also carried out a cell-based VEGF-A signalling blockade assay, where the cells were incubated with increasing concentrations of antibody in the presence of an EC90 concentration of recombinant VEGF-A for 6 hours before luminescence was measured. Titration of increasing amounts of antibody prevents VEGF-A from binding to its receptor, which in turn activates a signalling pathway within the cells of the assay, which results in a luciferase expression and thus luminescence. The EC50 concentration of antibody that induces a response halfway between the baseline and the maximum response measured after a specific exposure time was found to be the same as commercially available Bevacizumab biosimilar that was produced in mammalian cells. One of our recent customer projects also involved the production of an antibody, but this time for diagnostic use in a lateral flow assay. The mouse monoclonal IgG1 antibody was produced using a very similar pipeline to our Bevacizumab biosimilar, resulting in process yields of approximately 180 mg per kilogram of leaf tissue. Isoelectric focusing analysis of our antibody compared to a hybridoma version showed slight differences in antibody PI, likely due to differences in glycosylation pattern, which was consistent across all 3 batches produced here at Leaf. Glycosylation differences can be expected compared to hybridoma cell lines, as plants can carry out different glycosylation modifications on proteins. However, plant lines can be engineered to produce humanized glycosylation patterns if this is required for the therapeutic or diagnostic protein of interest. Despite these differences, activity analysis of the plant-made antibody within the intended lateral flow assay saw an identical activity profile when compared to the hybridoma-produced antibody, suggesting that the differences in glycosylation had no impact on the functionality of the antibody